Welcome to episode 1 of Epigenetics Explained. In this video, we will be covering the basics of DNA, RNA, genes, chromatin, histones, and epigenetics. Don't worry if these words sound foreign or intimidating. I will try to explain them in as simple terms as I can. First, we need to understand what DNA and RNA are. Most of you probably know that DNA is that all-important molecule that encodes our genetic information. But what does that even mean, and how does it do this? Let's start with the name itself. DNA stands for deoxyribonucleic acid. To understand its components, it can be helpful to think of DNA as a ladder. The backbone of our DNA ladder is made up of sugar molecules called deoxyribose, hence the first part of the name here. So these are the deoxyribose molecules, and these molecules are joined together by phosphate groups, which are these. Deoxyribose has a 5-carbon ring structure, meaning that it has 5 arms of carbon atoms. These so-called arms are numbered 1', 2', 3', 4', and 5. Oh, can't see that. 5'. And these are numbered in a clockwise direction. Phosphate groups connect the 3' of one deoxyribose molecule to the 5' of its adjacent molecule. Therefore, one end of the whole DNA molecule will have a free 5' carbon, which is here, and the other end will have a free 3' carbon, which is here. The nitrogenous bases, which are where our genetic information is encoded, form the rungs of our DNA ladder. There are 4 different bases, or letters if you will. Adenine, which always pairs with thymine, and guanine, which always pairs with cytosine. Cytosine is particularly important when it comes to epigenetics, so make sure to take a note of that. Now, RNA stands for ribonucleic acid and is very similar to DNA. There are only 3 main differences. The sugar molecule in RNA is called ribose instead of deoxyribose. One of the bases is called uracil. Now this is the equivalent of thymine in DNA as it always pairs with adenine. And RNA is single-stranded, so it's like a ladder but chopped in half vertically. Now that we've covered the molecular components of DNA and RNA, let's zoom out a bit. Literally and metaphorically. DNA is divided into sections called genes, and these code for a specific functional product. They are usually 10 to 50,000 base pairs long, and a base pair is the equivalent of 1 rung on a DNA ladder, which would be this. And a nucleotide is similar, though it refers to the rung and the backbone, including the phosphate groups and deoxyribose molecules, whereas a base pair only refers to the rung. Contrary to how it is usually depicted, DNA doesn't exist as its own isolated molecule. It is wound around clusters of proteins called histones. The reason for this is to make it easier to compact DNA into structures called chromosomes so that the cell can divide efficiently. Histones are positively charged, which is what allows them to stick to negatively charged DNA. Chromatin is the combination of DNA and histones and is made up of units called nucleosomes. Each nucleosome is made up of 146 DNA base pairs coiled around an octamer of 8 histone proteins. These 8 histone proteins include 2 of each core histone, which are H2A, H2B, H3, and H4. Here is another diagram of a nucleosome. Although it looks more complicated, the idea remains the same. There are 2 of each of the 4 core histones to form a histone octamer, around which DNA is wound to create a nucleosome. A loose open arrangement of chromatin is called euchromatin, and this promotes gene expression. On the other hand, a tight closed arrangement of chromatin is called heterochromatin. This restricts gene expression. Chromatin can be compared to beads on a string, or more accurately in my opinion, thread on a sewing bobbin. Now onto the exciting part, epigenetics. Before I even tell you what it means, let's deconstruct the word itself. Epi is Greek for above or on top of, and genetics obviously refers to genes. So the word means above genes. This term was coined by Conrad Waddington, who also came up with this definition. He says, "It is the causal interactions between genes and their products which bring the phenotype into being." Now, the phenotype is the collective physical traits of an organism. The definition of epigenetics has evolved over the past few decades, so this definition would today refer to the central dogma of molecular biology, which we'll get into in the next episode. Here is what might be considered a more modern definition of epigenetics: the study of heritable changes in gene expression that occur independent of changes in the primary DNA sequence. In other words, epigenetics is the study of changes that take place above the gene, which control the expression of certain genes. In future episodes, we will discuss what these changes are, how they affect gene expression, and their consequences at the genetic and organism level. Thank you for watching the first episode of Epigenetics Explained. I hope it has been useful in building a basic understanding of genetics and epigenetics, and see you in the next episode.